Welcome to Biochemistry of Metabolic Processes. Today, we are diving into redox systems. If you have ever wondered how the food you eat actually turns into the energy that powers your muscles and your brain, you are in the right place. We are going to build this from the ground up, so do not worry if you are completely new to this. Let us get started. Now that we know what we are studying, let us start with a simple analogy. Imagine a relay race, but instead of a standard baton, the runners are passing a bundle of pure, concentrated energy. In the microscopic world of your cells, this high-energy baton is an electron. When a molecule holds this electron, it is full of potential energy, much like a tightly compressed spring waiting to be released. Passing this electron from one molecule to another is the fundamental way your body moves energy around to keep you alive. If a molecule gives the baton away, it loses that energy packet. If it receives the baton, it gains that energy. Building on our relay race analogy, let us look at the formal scientific terms. In biochemistry, a redox system refers to a paired chemical reaction where oxidation and reduction occur simultaneously. The term redox is simply a combination of these two words. Oxidation is defined as the loss of electrons, while reduction is defined as the gain of electrons. Because electrons cannot just float around freely in the watery environment of the cell, one molecule must donate the electron, becoming oxidized, while another molecule immediately accepts it, becoming reduced. They always happen together, just like a baton must be handed from one specific runner directly to another. Since we understand that electrons must be carefully handed off, we need to introduce the specialized runners in our cellular relay race. In biological systems, these electrons are very often transferred along with a proton. This means we are essentially tracking the movement of entire hydrogen atoms. To handle this safely, your cells use specialized molecules like NAD+ and FAD. These act as dedicated electron carriers. Their entire job is to shuttle these high-energy electrons from the food we break down to the cellular machinery that manufactures our energy. They are the vital transport vehicles of the microscopic world. So, why do these electron carriers matter so much to your survival? Without redox systems, the food you eat would be completely useless to your body. When you consume a bowl of pasta, your digestive system breaks it down into glucose. But your cells cannot use glucose directly to power immediate tasks like flexing a muscle. Instead, your cells strip electrons from the glucose molecule through a series of redox reactions, capturing that energy in carriers like NADH. These carriers deliver the electrons to the mitochondria, powering the creation of ATP, the actual usable energy currency. If this stops, cellular death follows in minutes, which is exactly how cyanide poisons the body. Now that we understand the biological importance, let us look at the math that governs this flow of energy. This first formula calculates the standard reduction potential difference, denoted by delta E0 prime. Conceptually, this tells us how strongly electrons want to flow from one molecule to another. The symbol E0 prime A represents the standard reduction potential of the molecule receiving the electrons, measured in volts. The symbol E0 prime D represents the potential of the molecule giving away the electrons. In plain English, the total driving force is the difference between how badly the acceptor wants the electrons and how tightly the donor holds them. A large positive value means electrons flow eagerly. This brings us to our second formula, which is one of the most important equations in biochemistry. It connects the electrical potential we just discussed to the actual thermodynamic energy available to do cellular work. The symbol delta G naught prime represents the standard Gibbs free energy change, telling us how much usable energy is released or consumed. The variable N stands for the number of electrons being transferred, which is usually 2. The symbol F is Faraday's constant, acting as a conversion factor between voltage and energy. Finally, delta E naught prime is the voltage difference. If the voltage difference increases, the amount of energy released also increases proportionally. Before we move on, let us see this with a concrete example. We will calculate the energy change for transferring electrons from NADH to oxygen. We are given the potential for oxygen as positive 0.82 volts and NADH as negative 0.32 volts. First, we find the voltage difference by subtracting the donor from the acceptor, giving us 1.14 volts. Next, we plug this into our Gibbs free energy equation. We multiply negative 2 electrons times Faraday's constant times our voltage difference. This yields roughly negative 220,000 joules per mole, or negative 220 kilojoules per mole. This massive negative value means a tremendous amount of energy is released. So we have successfully used this formula, but let us take a moment to prove why it guarantees energy release. What this chunk of proof is establishing is the mathematical certainty of spontaneous reactions. We know that a positive voltage difference indicates that electrons naturally want to flow. Look at the Gibbs free energy equation. The number of electrons and Faraday's constant are always positive values. Because of the negative sign permanently built into the front of the formula, plugging in a positive voltage difference mathematically forces the final free energy to be negative. In thermodynamics, a negative free energy proves that the reaction is exergonic, meaning it releases energy for the cell to use. Now that we have proven how the math works, let us look at a smaller, more realistic single step within the cell. Instead of dropping all the way to oxygen, NADH donates electrons to coenzyme Q. The potential for coenzyme Q is positive 0.04 volts. We subtract the NADH potential, giving us a much smaller voltage difference of 0.36 volts. We plug this new voltage into the Gibbs equation, multiplying by 2 electrons and Faraday's constant. The calculation yields roughly negative 69.5 kilojoules per mole. This shows that the electrons are taking a smaller fall in energy, releasing a manageable amount of power. This smaller energy calculation leads us to an important biological concept. The result perfectly illustrates how the electron transport chain functions like a staircase rather than a single steep cliff. If the cell dropped electrons directly to oxygen all at once, the massive burst of energy would be lost as dangerous heat, potentially destroying the cell. By releasing the energy in smaller increments, like the 69.5 kilojoules we just calculated, the cell can efficiently capture it. Since it takes about 30 kilojoules per mole to make one ATP molecule, this single step releases more than enough energy to contribute significantly to ATP synthesis without overheating the system. Let us visualize everything we have learned by looking at this diagram of the redox tower. On the vertical axis, we have the standard reduction potential in volts, starting negative at the top and becoming highly positive at the bottom. Notice the staircase structure. At the very top, we see NADH holding the high-energy electrons. As the yellow arrows show, the electrons fall down the steps, first to coenzyme Q, then to cytochrome C, and finally landing at oxygen at the bottom. Each downward step represents a positive change in voltage and a release of free energy. This diagram beautifully captures how the cell breaks a dangerously large energy drop into safe, manageable steps to synthesize ATP. We have covered a lot of ground today, so let us reinforce the most important ideas. First, redox systems are the biological equivalent of a controlled waterfall, passing electrons to capture energy. Remember that oxidation is the loss of electrons, and reduction is the gain. We learned that the reduction potential difference drives this flow, and the Gibbs free energy equation allows us to calculate the exact amount of usable energy released. Finally, we saw how cells use a staircase cascade to safely harvest this energy to make ATP. You now understand the fundamental mechanism behind cellular respiration. Great job, and keep exploring the amazing chemistry of life.